Well, welcome everybody, and thank you for joining us this afternoon. We're quickly approaching the end of this year, which has been quite an unusual and a challenging year. But with vaccines on the way, the outlook for next year is beginning to look promising. And Brian and I were just talking off camera saying, yeah, there's hope at the end of the tunnel. It's going to be a rather long tunnel, but there is hope. We'll certainly see what happens with the direction of the virus over the next few weeks to see if our plan to offer limited in-person classes beginning in third term will be possible, but we'll have more to come on that in the coming weeks. We'll keep our fingers crossed. And looking forward further, we're more hopeful that by fall of 2021, things might be back to normal or some sense of normalcy, and we'll be able to have more people return to the school. I know I'm eager to get back to the building for in-person activities, and I'm sure you are as well. But we just need to hold on a little bit longer as we've been doing these many months, and we'll continue to get through this pandemic together. So as we turn towards the holidays, I want to make sure that before we begin today's program, I wish everyone a safe and a very healthy holiday season. And I hope everybody gets a chance to unwind and decompress and enjoy the holidays. But today we're gathered together to celebrate Brion Maher and his promotion to full professor. As you know, earning the rank of professor represents an incredible achievement and confirms not only the respect and high regard of the school, but also of colleagues across the nation and certainly throughout the world. We are thrilled to host this Dean's Lecture honoring Professor Brion Maher and to learn more about his work. Brion is an accomplished scholar and internationally recognized in psychiatric and behavioral genetics. His research focuses on the genetics and epigenetics of psychiatric disorders, primarily developmental and behavioral disorders, as well as methods development for genetic epidemiology of complex diseases. He has made substantial contributions to large-scale studies requiring cross-disciplinary collaborations, as well as multi-site consortia. He has published over 150 publications, many, if not most, in high-profile journals. And very importantly, several of his publications are authored by his direct trainees. Brion has a strong record of service to his research community and is an outstanding citizen of the school and the Department of Mental Health. He is committed to teaching and advising, and with Dr. Renee Johnson, Brion co-directs the NIH NIDA-funded Drug Dependence Epidemiology Training Program, providing rigorous training in research on substance use and substance use disorders from a public health perspective. This program, without a doubt, is producing the next generation of substance use epidemiologists. Now, before coming to the Bloomberg School, Dr. Maher held faculty positions at both the University of Pittsburgh School of Dental Medicine and Virginia Commonwealth University, where he was a member of the Virginia Institute of Psychiatric and Behavioral Genetics in the Department of Psychiatry and Human Genetics. In 2010, we were very fortunate to recruit him to the school after a national search, and he was brought to the Department of Mental Health as an associate professor and then promoted to full professor in 2017. He holds a joint appointment in the Department of Psychiatry and Behavioral Sciences in the School of Medicine. Brion received a BS in Biological Sciences and an SCM and PhD in Human Genetics, all from the University of Pittsburgh. I am so delighted to introduce Professor Brion Maher, and I look forward to his lecture. And with that, I'm going to turn it over to Brion.
Thank you, Ellen, for the very kind introduction. I'm very honored to be giving this talk. Obviously, a bit sad not to be seeing everyone's faces, although some of you may make me nervous, so maybe it's a good thing. Before starting, there were two things I want to mention. I have an acknowledgement slide at the end of the talk, but I want to acknowledge up front that everything I present is the result of long collaborations at the institutions that that Dean McKenzie mentioned, and I've had the incredible enriching experiences at those institutions, and I'm thankful for the many colleagues over the years who've become good friends with whom all of this work would have been, frankly, very boring. And then the other thing I want to mention is that I use the term addiction in my title, and what I'm really going to be talking about are substance use and substance use disorders. But including that in the title of the slide seemed like a bit of a mouthful. So a brief outline of the talk that I'm going to give today. First, I'm going to talk about the population, the impact of substance use and substance use disorders at the population level. I'll discuss how substance use disorders are defined both in clinical practice and in etiologic research. I'll provide a brief overview of the current state of substance use disorder, the substance use disorder genetic literature, and I'll provide a more in-depth summary of our own local work, including single gene and polygenic associations with substance use disorder, both in genetic and epigenetics. And then lastly, I will discuss our ongoing work in the epigenetics of substance use disorder in 2 local cohorts. And finally, I'll provide a wrap up. And I want to give the conclusion on the first slide because I think it helps to shape the way that you listen to the things that I talk about. And my general point is that the substance use disorder diagnosis as it's currently constructed and used clinically does not adequately capture risk across the lifespan for many substance use disorder associated outcomes, and that I argue for a continuous liability approach. That continuous liability approach will include the fixed and dynamic risk and indexed by the genome, but moreover will identify those not just currently suffering from a substance use disorder, but those at risk for a substance use disorder who are a uniquely at-risk population. And so jumping into substance use disorders in public health. This is from the Global Burden of Diseases. This was the last detailed analysis of the substance use disorder findings from the Global Burden of Disease, although it's published annually. I included this as a reminder of multiple things about the public health consequences of substance use. Substance use accounts for a substantial proportion of the disability adjusted life years worldwide. And you can see alcohol accounts for about 4.2% and drug use about 1.3% worldwide. But I'm also showing this to remind that the burden of drug use is higher in North America relative to other causes versus the rest of the world, especially among drug use, with alcohol accounting for 4.5% and drugs accounting for 5.1% of disability adjusted life years, respectively. So clearly, substance use disorders are a clear health concern and public health concern. The factor underlying this that drives this is this, in the US, this persistent high level of substance use disorders in the past year. So what we see here are data from the National Survey on Drug Use and Health administered by SAMHSA. You can see that over the past 18 years, we've had this persistently high rate of substance use disorders in the past year among adults in the range of 8 to 9%. There's a discontinuity here between the graphs, and that is owing to the change in diagnostic criteria from DSM-4 to DSM-5, but the lines with the arrows pointing to them are pretty much equivalent. Substance use disorder in the past year in adults across across alcohol and all drugs. So to me, the important point is that those with substance use disorders are a vulnerable population. There's a pretty significant baseline prevalence of this problem. And I think substance use disorder is an important marker that's an underutilized marker in the time of crisis. And this has been evidenced by 2 of the most recent public health crises. The first one is the opioid epidemic. Many of us have seen the slides of this massive uptick in opioid overdose over the years starting in the early parts of the teens and peaking relatively recently. But this is a graph of past year heroin use as indicated by the NISDU. Over this period of time, we experienced this massive increase in opioid overdose. But what you can see is there are only moderate upticks in heroin use in that time in the 18 to 25 and the 26 plus age group. Doesn't really seem commensurate with the increase in overdose. But what I would argue is that this modest uptick in heroin use challenged the drug supply chains, led to substitution and adulteration of the supply, and a consequent public health crisis. And but the important point is that this relatively subtle change in the environment had a massive, disproportionate public health consequence in this vulnerable group, highlighting the need for understanding factors underlying persistent substance use disorders. And this is a very recent evidence of the almost unique vulnerability of those living with substance use disorders. Using electronic health records in about 70+ million patients in the US, these researchers, including NIDA Director Nora Volkoff, demonstrated substantial increased risk for COVID infection among those with a recent substance use disorder. So this is important. This very pronounced risk was among those with a substance use disorder diagnosed in the 12 months prior to the onset of the pandemic. You can see an almost 8.7 increased risk among those with any recent substance use disorder, and then the risk was especially pronounced with those with a recent opioid use disorder. Again, people with substance use disorders are among the most at risk for significant morbidity and mortality in public health crises. I jumped right into talking about substance use disorders without first defining them. So let's tackle that now. This table is drawn from the paper cited below. It depicts the change in the diagnostic criteria for substance use disorders from the Diagnostic and Statistical Manual of the American Psychiatric Association from the 4th edition to the 5th edition. Several important, and this was 2014, 2015. Several important changes from 4 to 5. The first is that there were separate diagnoses for substance abuse and substance dependence in previous iterations. So those were merged into a single, in DSM-5, a single substance use disorder diagnosis. Other important changes were that the legal problems, which is largely a social construct, the legal problem symptom was dropped, but an important symptom was added, and that is drug craving. Importantly, this allows for a severity scale with 2 or more symptoms considered mild substance use disorder, up to 6 or more considered a severe substance use disorder. And this scale can be administered for any of 10 specific drug classes listed below, or unknown or other drug classes. This is it's an empirically driven clusters of symptoms. It's a step in the right direction. But for those studying the etiology and course, and especially those of us studying the genetics of substance use disorders, it does not eliminate all the head scratching that comes with constructing a phenotype or trait for study. And so this slide is credit of a good friend and colleague, Professor Michael Vanyukov. And so when geneticists tend to think about disorders or traits, we crave something that's really trait-like, that maximizes the heritability or proportion due to genes, and might be reflective of the biology. And for that reason, DSM diagnosis has historically been lacking. So remember from the previous slide that a DSM diagnosis can be reached with 2 or more symptoms of 11 total symptoms. So in DSM-5, if we sum across 11, choose 2, 3, 4, the number of possible symptom combinations by which someone can have a substance use disorder is 2,036. So 2,036 unique ways in which someone could be diagnosed with a substance use disorder, and that is per substance. So while the symptoms are useful, the heterogeneous nature by which this discrete disorder can be reached is less than ideal. And this is highlighted by this quote by former NIH director, Tom Insel. Unlike our definitions for ischemic heart disease, lymphoma, or AIDS, the DSM diagnoses are based on consensus about clusters or clinical symptoms, not any objective laboratory measure. And for those of us studying the biological basis of something, that leaves a little bit to be desired. Moreover, DSM diagnoses refer to the prior 12 months, and it's really blind to the stages of substance use from initiation through continued use, abuse, and then dependence. This trajectory is especially important to think about with respect to etiologic research, where different factors may have differential impact at different times in the course of the disease. And I'll revisit this slide several times, adding additional information. So a unique feature of substance use disorders relative to most other diseases or disorders is the requirement for some exposure for development and diagnosis of the disease. You can't be diagnosed with a substance use disorder without exposure to the substance. So a parallel example to this is phenylketonuria. It's an autosomal recessive disorder due to mutations in phenylalanine hydroxylase. the reduced activity of the enzyme leads to accumulation of phenylketone. Historically, the phenotype had been intellectual disability or seizures, and the treatment is just simply removal of the amino acid phenylalanine from the diet. In the current era, we are in an environment that's equivalent to E4 in this slide. We have nearly ubiquitous testing for PKU in newborn screening, and identification of a new case allows immediate treatment by an environmental modification. And the environmental modification is removal of phenylalanine from the diet. And phenotypically, this essentially cures the disease. The IQ average IQ is normal, and and seizures are not present. And so the question is PKU still heritable? Well, the answer is not really definitive, and it really depends on whether or not you accept that the phenotype, this phenotype that's displayed on the screen is the phenotype of PKU, or the genotype present is the phenotype of PKU. And so I don't want to give the impression that substance use disorders are some nebulous group of symptoms, but much like the metabolism of PKU, there are biological differences in an individual's response to ingesting a particular chemical. And much of the work of addiction biologists seeks to really understand the common mechanisms underlying drug metabolism, and then the role of inter-individual variation in that drug metabolism. And that's really where genetics come in, that individual variation. So this presented here is is a well-developed theoretical framework of the biology of addiction, published by the NIAAA and NIDA directors, Koob and Volkoff, that is based on that provides a theoretical framework of the biology of addiction based on decades of data from preclinical and human studies. This is maybe a complicated looking model, but it includes the brain regions thought to underlie the cycle of addiction, from heavy use to withdrawal, to then anticipation of subsequent use, and includes the brain regions that are thought to underlie those. It also includes the behavioral components that are measurable, that drive that: exaggerated incentive salience, exaggerated reward deficit, and compromised executive function. And so much of the work around the behavioral dimensions in substance use disorder would focus on that. But importantly, this is a model of the brain mechanism after exposure and recurrent use, and does not capture the entire disease trajectory or the premorbid biological risk. So this is essentially a model of the brain's response to use and recurrent use. So revisiting this slide, for those of us interested in the genetics of substance use disorders, where do we start? So I've marked in the approximate parts of this trajectory that most studies consider control in cases. So this is starting with initiation, first use of a substance through use, continued abuse, eventual dependence, on to cessation from the substance. And typically, studies will include as controls those who've initiated or not initiated, or initiated, and those who've used and not become dependent. And cases will typically be those with a use disorder, abuse, or dependence. Now, there will be, certainly studies that define it more specifically, but these may be the most common categorizations of case control. As I mentioned before, we have this well-formulated model of the neurobiology addiction of after exposure, but as you can see, that model only would capture this part of from use through abuse and dependence, and not those who haven't initiated. So why does this matter? Because the conclusions we can draw from different studies will really be based on the decisions we make in sample ascertainment. So for example, studies of the variation among drug users here will not inform studies of prevention. Studies including controls who have never used will not inform drug-specific liability, and so on. So I want to argue in favor of one approach of defining substance use disorders that can fit well into the current substance use disorder measurement models, and can capture most of the prior work on substance use disorders. This is largely the work of a longtime colleague, mentor, and friend, Michael Vanyakoff at the University of Pittsburgh. So instead of a dichotomously defined disorder, it's likely that substance use disorders are best conceptualized as a phenotype that varies as a continuous liability distribution that is normally distributed in the population, where the probability of developing a substance use disorder increases as you move to the right on this liability distribution. And then above some threshold, the probability of developing a substance use disorder approaches 1. The nice thing about, a couple of nice things about this, it's liability distribution can be made up of multiple domains of measurement, including non-drug use behaviors, environmental factors, and biological measures. Importantly, given complete information, an individual's liability phenotype can be measured at any point in the substance use disorder trajectory, even before use or after complete cessation of use. And I believe it is inherent that, that genes influence this risk and should be included in liability for substance use disorder measures. Another important point is that an individual's liability is dynamic. It can change throughout the lifespan with experiences. So peer and family influences, for instance, can positively or negatively impact liability. Adverse life events could negatively impact the liability and move someone closer to the threshold of a substance use disorder. Initiation of substances can push the trajectories to the left or the right. And for instance, an unpleasant initial experience can decrease the likelihood of subsequent use. A well-known example of this is the flushing response that some experience with drinking alcohol. So and that would be sort of shifting the liability to the left or decreasing someone's probability of developing a substance use disorder. So the strengths of this liability concept for measurement of substance use disorder risk include that it captures the mechanisms and biobehavioral characteristics that pertain to the entire course of the disorder and changes in that risk. And it overlaps with the existing psychological and psychopathological constructs that have been used to measure addiction for years. And it need not be mostly biological or purely biological or mostly genetic or purely genetic, but it is useful to include some biological measures in liability constructs. Okay, so I'm gonna move now to the genetics of substance use disorders and a little bit about what we know so far. So why genomics in public health? I think it's important to acknowledge the largely a shameful history of the application of genetics for public health before the 1960s and 70s globally, and to always remind ourselves that when we're pursuing genetic studies in public health, that we should really ask ourselves the question, why? Why are we doing this? And is it really in the benefit of the public? And so I would argue that we can improve health by improving our understanding of all contributing factors to any disease, especially if those factors indicate a risk for a group of disorders, such as those with the significant public health impact of substance use disorders, as we've seen, as I mentioned earlier, in the 2 most recent public health crises, including the one we're in the middle of. Additionally, we may improve treatments by understanding the genetics of substance use disorder. For instance, it may be that some patients will benefit from targeted behavior change, while others may benefit from precision medicine with regard to medication-assisted treatment. Lastly, and I think this is important, we're faced with a reality where millions of Americans already have access to their genomic information through commercially available genotyping platforms, and many of those companies are providing risk assessments to patients direct to consumer. I think it's incumbent upon us to ensure that appropriate information is available to allow the public to appropriately understanding the meaning of these genetic tests. If we accept that something is worth studying the genetics of a trait, first question geneticists typically ask, is it heritable? This is a slide from a 15-year-old review paper, but there haven't been many twin studies since, and what it shows is that for addiction and related phenotypes, the heritability remains between 40 and 60%, and that heritability would, the best definition is the variation in the trait that's due to genes. Moreover, this seminal work has shown that the genetic risk for substance use disorders is largely shared across drug classes. So this is a path diagram representing the results of a twin model across 5 commonly used substances. The bubbles up top represent latent genetic factors, and the numbers in the lines represent the strength of correlation. So for instance, the two genetic factors are correlated with each other, 0.82, or standardized regression coefficients of the relationship between the variables. And you can see the significant path coefficients from the genetic factors to the symptomatology of the specific substance use disorders. It's also, so this up top, these common liability, these cross substance genetic factors are commonly referred to as shared or common liability. These bubbles below that point to one substance, those are the substance specific genetic factors. And you can see those also have pretty large path coefficients. And so what this is demonstrating is both common across drug classes, shared genetic liability, and then drug class specific genetic liability. And so there are two classes of substance use disorder risk genes that we really need to worry about. This is a cartoon version of the same thing, with the common genes above represented and the G's below representing the drug specific genetic influences. And what you might imagine is that drug specific genetic influences include things that are, for instance, in the metabolic pathway of that drug. So how the drug is, specific drug is metabolized, where the common genes are the common metabolic pathways, behavioral pathways, the pathways in the reward pathways, the genes involved in the reward pathway in the brain. So revisiting this slide again, thinking about the impact of when these common and specific genes are impacting substance use disorder risk from initiation, we can see larger on the environment, through use, abuse, dependence, and cessation. Clearly the role of common liability genes can act early, for instance, through novelty seeking, which is a heritable behavioral trait, and also later through genes influencing brain reward mechanisms, while the substance specific genetic effects can't really be observed, can't really be observed until they're after sufficient exposure. So you will not, the gene influencing, for instance, the gene specific to alcohol use disorder will not be, their impact will not be realized until there's sufficient alcohol exposure. One other point, there's a growing interest and a particular interest in our group of the relative role of genes and environment on cessation and long-term treatment success. So to summarize so far, substance use disorders, while defined as a clinical syndrome, are really lacking an adequate measurable biological definition. It's a multi-stage disorder with an obligate exposure, an accepted neurobiological model, and a diagnostic framework that fits well within this would fit well within this continuous liability metric. The solution in the short term is really to pursue multiple modes of investigation. So I will present recent work that we've been involved in that pursues multiple modes of investigation. Although the long-term goal of the field is really to fill in the gaps for the factors accounting for variation at each of those stages that I mentioned for common liability and drug class specific liability. So to date, there have been multiple replicating findings from genome-wide association studies for substance use disorders. I'll talk a little bit more about what a genome-wide association study is on the next slide. These are largely the result of large consortia, for instance, the substance use disorder group of the Psychiatric Genomics Consortium. The early successes, the successes to date have largely been drug class specific and in the metabolic pathways of the drug. So for instance, for nicotine dependence, a cluster of nicotinic receptors are the largest genetic association signal. For alcohol use disorder, aldehyde and alcohol dehydrogenase genes are the largest signals. For caffeine dependence, cytochrome P450 enzymes, including CYP1A2 is the largest signal. And then there are, I'll call them emerging successes for opioid use disorder and cannabis use disorder. Those samples just aren't quite as large yet, but as they sample sizes grow, we expect to have similar successes. And I'm going to present some specific examples from the substance use disorder PGC group. So this is the first effort of the substance use disorder group of the Psychiatric Genomics Consortium. This was led by Raymond Walters at the Broad Institute and a very long list of authors. It's a genome-wide study of alcohol dependence in nearly 15,000 cases and 38,000 controls. This graph here is a Manhattan plot. We call it a Manhattan plot. And it's our typical approach to displaying the results of a genome-wide association scan. And so that's a genome-wide association scan is a test for case control differences in usually an allele frequency across millions of markers spread across the genome. So on the x-axis here, we have the location of those variants across the genome. And on the y-axis, we have the minus log 10 p-value for a test of the case control differences in allele frequencies at those particular polymorphisms. And this red dashed line represents genome-wide significance. So because we're performing so many tests, we have to account for multiple testing. Many people use a Bonferroni approach. So our target p-value is something like 1 times 10 to the minus 8, 1 times 10 to the minus 7 in that range. You can see there's a pretty large signal here, and that's the gene I mentioned previously, ADH1B, that's involved in alcohol metabolism. The second is a brain expressed gene, this small signal here. There's not a lot of supporting SNPs around it, so that's yet to be replicated. One advance as a consequence of these large studies, so this draws from on the same study, is that we have large studies with genetic variants measured across the genome. And this gives us the ability to create composite metrics of genetic risk for a disease, or to compare the genetic risk for 2 particular diseases and estimate their genetic overlap or genetic correlation. And so as can be seen here, the genetic risk for alcohol use disorder is pretty strongly associated with cannabis initiation, smoking initiation, nicotine dependence, smoking cessation, alcohol consumption, and in multiple alcohol consumption scores. Smoking cessation, it's negatively correlated. And so what this is reflective of is this concept of common liability, that the genetic risk for alcohol use disorders is strongly correlated with the genetic risk for other substance use disorder outcomes. This was another effort of the PGC on cannabis use disorder in 20,000 cases and 360,000 controls. These results are a little more modest with 2 genome-wide significant findings. But I think what's more interesting here is the set of findings, as doing the genetic correlations as I previously discussed, of cannabis use and cannabis use disorder with other traits. And so note these significant positive correlations with the psychiatric disorders here in green and the substance use traits down here in, I guess that's pink or purple. And this reminds us of not only the common liability, but also the polygenic architecture and complex behavioral contributions to substance use disorders. So substance use disorders aren't happening in a bubble, and these strong genetic correlations with, for instance, a very strong premorbid risk factor like ADHD are not surprising. So I just mentioned an advance being that we can create these composite genetic risk scores. One of these approaches is called polygenic scoring. This is a really nice schematic of polygenic scoring. This approach generally relies on a very large external genome-wide association study, an estimate of the, so relies on a large external genome-wide association study. From that large external genome-wide association study, you explain the combination of genetic variants and the weights, usually log odds ratios or beta regression betas, and then create in a novel test data set individual level genetic risk scores. And these, as I mentioned, are typically weighted sum scores. And then test for association between that genetic risk score and some phenotype in your target data set. It's also important to note that there are many approaches that are much more sophisticated than just weighted sum scores. People here and elsewhere are working on those, including us. But this is a relatively straightforward approach and something that's been widely used. And if it works very well, as it does here for the PGC schizophrenia sample, from a GWAS of a couple 100,000 subjects, you can generate information that will allow you to create pretty useful individual level risk metrics. So as you can see here, individuals in the top decile for schizophrenia genetic risk are upwards of 20-fold, have an upwards of 20-fold overrepresentation in case versus control samples. And I say it that way because that's literally what this is. So the caveat is that these findings really need to be validated in external studies and in the population and not just in these ascertained case control samples. But nevertheless, this indicates that GWAS to an extent is really able to capture a meaningful proportion of the genetic variation in traits. And if we indeed have polygenic scores that can, that those in the upper decile have a 20-fold increased risk for a disease outcome, that those types of measures will be absolutely clinically useful. So we've done a fair amount of polygenic score work. I'm gonna breeze through this part, largely in the PERC cohorts. This is the Prevention Intervention Research Center cohorts. The current PIs are Nick Ialongo and Bill Eaton recently. In the past, Jim Anthony and Shep Kellum. These studies in the 80s and 90s enrolled elementary school students in first grade in a universal intervention and followed them into young adulthood. We have genotypes on about almost 1,350 of those participants who also have been assessed on substance use throughout adolescence and into adulthood and even into approaching midlife. And so we've been able to look at things like, so let me state, this is a sampling. There are a few more papers that I'm not going to highlight here. This has largely been the work led initially by my colleagues Rochelle Musey and more recently by Jill Rabinowitz. What we've been able to look at, for instance, are the genetic risk for externalizing symptoms, association with marijuana use in the PERC, the genetic risk for internalizing disorders and antisocial personality on alcohol use in the PERC, the polygenic risk for a major depressive disorder from the PGC on internalizing symptoms in the PERC, conduct disorder genetic risk on peer report of bullying behavior, smoking genetic risk on age of first tobacco use, and conduct disorder genetic risk on risky sexual behavior. And so I'm going to provide a brief summary. There's a lot of work to be done in polygenic scores. They provide a useful framework for a metric of risk. They exploit the large amount of work that's been done in the field. So we're maximizing that investment of the funding agencies. But much work needs to be done to ensure appropriate interpretation and clinical application. As I mentioned previously, folks are able to download their data from the commercial genotyping source that they've used and upload it to other sites and calculate their polygenic risk for a whole host of outcomes. And I think we owe it to the public to create and disseminate appropriate information so that those kinds of risk scores are more interpretable than they would be otherwise. So I'm going to transition now to epigenetics, and the rest of the talk will be on some work we've been doing in epigenetics. The original definition of the term epigenetics was proposed by Conrad Waddington in the 1950s, and he thought he said it was the branch of biology which studies the causal interactions between genes and their products which bring the phenotype into being. I think a more modern definition, and probably the one that's most applicable to what we're doing, might be something like the study of changes to the genome, but not the DNA sequence that lead to changes in gene expression. And you'll note that I didn't address reversibility or heritability or transmittability of those changes, because I think those are all open questions. This is kind of the hypothesis. It's the theoretical framework is something like this. Continued drug exposure leads to epigenetic changes that over time play a role in the reinforcing effects of drugs of abuse. So this is part of the liability distribution. It lays further on the right after exposure to drugs. And what I would propose is that that epigenetic markers could develop into biomarkers of stages of liability or metrics of liability. In terms of epigenetics, I'll primarily focus on methylation, which is the addition of a methyl group to the 5th carbon of the nucleotide cytosine. That usually occurs at a C that's preceding a guanine or a G, hence the term CPG, where the P is the phosphodiester bond between the nucleotides. Generally, it's assumed that methylation represses nearby gene expression. There are many interesting questions out there about methylation including the pace at which methylation is reversible. Those are not, I can't answer the questions on those, and I think those are active areas of investigation. But clearly, this work now can highlight important pathways in therapy. The goal of many epigenetic studies involves identifying locations in the epigenome where the DNA methylation is associated with a measurable variable of interest. It typically refers to comparing proportions of cytosines that are methylated at specific sites. That could be, a single site, it could be groups of sites in a region, or it could be genome-wide. And there are these common approaches like DNA methylation age that are used now. And methylation can be measured using array-based or sequencing-based approaches. Everything I'm going to talk about is measured using array-based approaches, specifically the Illumina EPIC array. And a study that examines across the epigenome association is called an EWAS, or an epigenome-wide association scan. When it works well. This is evidenced by a meta-analysis here of cord blood, maternal smoking versus not, and 6,073 CPGs were considered statistically significant after multiple test correction for differences at site-specific methylation in the cord blood of newborns. So when it works well. We've engaged in recent collaborative work in the live cohort. The PIs of that are Shruti Mehta and Greg Kirk in epidemiology. The ALIVE is a prospective cohort designed to characterize the incidence and natural history of HIV infection among those who inject drugs in Baltimore. And over 5,000 folks have been enrolled since 1988. And the assessment happens up to twice annually with biosampling when subjects visit. And we have a genome-wide SNP data on about 2,400 subjects and genome-wide methylation data on 1,200 visits from about 450 subjects. And they were selected for periods of use and abstinence from peripheral blood. And we also have longitudinal drug use data. This is the descriptive table for an EWAS subsample with methylation data. This is work led by April Hsu, a former doctoral student in my group and in the Department of Mental Health, who's now a postdoc at Columbia. This is an EWAS of HIV status in the ALIVE cohort. So we can think of this as a positive control where there's a clear biological difference between these groups. We performed the analysis in 397 subjects, a small sample, who were abstaining from drugs when the sample was collected. As you can see, the sample is mostly male, Black, and about 1/3 HIV positive. On the next slide, I want to point out we were able to replicate our finding in the same subjects, but in samples when they were injecting, to show that this is a finding that's consistent with HIV. And you can see this red line is epigenome-wide significant. So after correction for multiple testing, you can see that after we account for cell heterogeneity, technical covariates, ancestry, and so on, there are many CPG sites that are significant after correction. Again, this is like a positive control to show that the approach is working, that we're getting accurate measurements. It's a known discrete significant biological difference between the groups. And but I want to draw your attention to the top gene, NLRC5. The one above it's an open reading frame. That just means a sequence of DNA where there's a start codon but no stop codon, and the function of those is frequently under investigation. And so NLRC5 is located on chromosome 16q13. It's an intracellular pattern recognition receptor that's involved in innate immunity to viruses. It's a regulator of interferon activity. But interestingly, it's a known site of the integration of the HIV virus in the human genome. So further questions we have is this a non-random integration site? And does integration here impact disease course? So we've also repeated these similar analyses on drug use using linear mixed effects models, adjusting for to account for the fact that these are samples drawn from the same subject at different points in time when they're actively injecting drugs and when they're abstaining from all drugs of abuse. We've run 4 analyses for any drug, heroin, actively injecting heroin, actively injecting cocaine, and then frequency of use. And as you can see, we don't see the kind of whopping signals we see for HIV for this EWAS. It's a little bit disappointing. But one thing that we did was we looked at the intersection of the findings. And so when we look at the top 0.1% of those, we see about 24 sites, whether we look at the CPG or the gene, that are consistent across these. Now we have to recognize that these are highly correlated analyses. So this isn't like independently we happen upon these 24 sites. It's the same set of subjects and similar phenotypes. Nevertheless, the very interesting gene is among the top findings, and it's FKBP5. FKBP5 is interesting because there's a large amount of existing evidence on the role of it in stress response and of differential methylation in response to chronic stress and stress-related psychiatric disorders, for example, depression and PTSD. And it may be that injection drug use, the part of the biological signature of injection drug use is chronic stress response. And interestingly, much of this work on FKBP5 has happened here at Hopkins. I'm sorry. Okay. In an attempt to replicate the alive findings that I just presented, which was in peripheral blood, we examined methylation and gene expression differences in prefrontal cortex of opioid overdose cases versus group and psychiatric match controls. This is work done in collaboration with the Lieber Institute. The site PI was Andrew Jaffe. And methylation was measured using arrays and gene expression by RNA sequencing. And the analytic work has been largely done by Dave Sosnowski, who's a DDET, Drug Dependence Epidemiology Training Program postdoc and in my group. And this is the descriptors for the sample. Note that after receiving the final tox screens, 5 of the site controls actually tested positive for an opioid. A case status was determined by acute opioid intoxication as a cause of death. And so there were 5 folks who had a positive opioid tox screen, but not as a cause of death. We eliminated those folks from the analysis. This is the epigenome-wide association scan. Nothing that exciting. And so, you know, it was a little disappointing. Moreover, there was no overlap or minimal overlap, non-significant overlap with the findings in the live. So the brain epigenome looks with opioid exposure looks different from the blood epigenome with opioid exposure. But we also looked at gene expression differences by case control status by RNA sequencing. This is a volcano plot of the log2 fold change between cases and controls. And so on the x-axis, it's the log2 fold change. So anything on the left here is underexpressed in cases. And then this is the minus log 10 p-value. So what we see, this red dot up here, NPAS4 is a gene that's significantly underexpressed in cases and genome-wide significant. This gene NPAS4 is, as you can see, stands out among the crowd there and is interesting for a variety of reasons. It's an immediate early gene, which means it's a transcription factor, which is a gateway to transcription of other genes. And more importantly, in preclinical studies, it's differentially expressed with reward-related behaviors and acute cocaine and opioid administration. And so this is going to be, you know, a focus of a fair amount of work in the future for us. So the obvious question is what next? And what I would argue that we need are better measures of dynamic substance use disorder risk. Not last 6 months, not last year, not lifetime, but right now. And so Greg Kirk and Johannes Thrull and Jill Rabinowitz and I are working on some approaches to measuring, for instance, real-time impulsivity using smartphone assessment coupled with collection of biomarkers. These may be better markers for risk of other substance use disorder associated outcomes, for instance, sleep. And Adam Spear and I are working on those sorts of studies now. The other thing we need are just larger samples. So some of these interesting biomarker studies are relatively small samples. And so we need replication or larger samples. And we also need to refine those signals. And then lastly, integration across domain and organism. There's a large amount of data out there that someone needs to combine, and there are ongoing efforts, including those funded by NIDA and those being requested by NIDA to integrate those things. Some other conclusions: substance use disorders are heritable. Much of this risk is shared across disorders. And the important thing really is that the liability to substance use disorder is a useful metric for capturing the dynamic nature of substance use disorder risk. It's a better indicator of those at risk, captures behavioral, environmental, and biological dimensions of risk, and may help us to identify those folks who are especially vulnerable, for instance, during times of public health crisis. And GWAS are currently starting to bear fruit. We're learning more about the architecture, the genetic architecture, and epigenetic influences may play a key role after exposure. This is acknowledgements. So the hazard in making a list like this is omitting people. So thanks to the many other colleagues and friends who have lent me an ear or trusted me with their data over the years. Most importantly, let me acknowledge the people who really make it possible, family and friends, and last but not least, NIDA funding. Thanks.
Great. Well, thank you so much, Brion. That's a great talk and an amazing body of work. I'm encouraging people we have a few more minutes. And if you'd like to ask Brion a question, you can put it in the chat. Probably that's easiest, just to put it in the chat, or you can put it under Q&A. But maybe just to get started, you started touching on this in the very, very end, but I'd like you to talk a little bit more about how some of your work, the epigenetic work, for instance, and you talk about how it's going to help you better characterize the dynamic nature of substance use disorder. But can you talk a little bit about how that then might translate into clinical or service applications or public health applications? How is all this going to help us serve people?
Sure. So there is an expectation that as we transition through those stages from initiation to use, abuse, dependence, and ultimately cessation, that there are changes that there are changes that occur in the genome that are possibly reversible, that highlight the biological pathways involved in that risk trajectory, and that and not only highlight those that are involved in the risk trajectory, but also highlight those that may be important in successful cessation. And so the important thing is that with all this is that there is variability among those who progress to advanced stages of a substance use disorder. And I think understanding the biomarker differences, whether it's genetic or epigenetic or other, in those who are able to transition from a substance use disorder to cessation, may be helpful just in terms of the knowledge, but also highlighting the important biological pathways that could be targeted by next generation therapeutics. Yep,
great. So we do have a question from Bill Eaton, and I was going to give Bill and Shep Kellum a shout out. It was great to see the PERC data being used. I mean, it's just one of the great things I always marvel at is how people can use some of the cohort studies and the prevention studies that accumulate lots of data over many, many years can be used to answer new and novel questions. But anyway, Bill, you have a question. So your question is, does this work parallel the interest in so-called endophenotypes for schizophrenia?
I would say yes, certainly, a composite of the endophenotypes, whether those endophenotypes are FRMI measures, or those are behavioral measures, questionnaire-based behavioral measures, some other objective measures like pre-pulse inhibition, or some other, maybe I mentioned biomarkers already, or neuropsych, other neuropsych measures could certainly be used to create an endophenotype. And in as much as that the genetics are informative beyond the measurable phenotypes, then I would say that yes, that those for instance, genetic biomarkers would make sense for them to become part of the liability distribution, right? So I think the liability distribution is especially useful when we can't measure that risk either pre-morbid or during the course.
Great. Well, it looks like we're just about out of time unless someone has one last burning question. And Danny's chiming in. Nice job, Brion.
Thanks, Danny.
But it was a terrific talk, Brion, and as I said, an incredible body of work, and we look forward to
Thank you.
Learning more.
And you mentioned the value of these cohorts.
Yeah.
It's been a key to my and other people's success here recently.
Yep.
So, yeah.
Yep. No, it's terrific. And the people who start those cohorts should feel good about that. Because it continues to contribute to new discoveries.
There was another question. Do your studies look at the effects of adverse childhood events on the epigenome? Adverse childhood events were recently measured in the ALIVE cohort retrospectively. And so we're in the process of looking at the association between those measures of adverse childhood events and epigenetic signature in the ALIVE cohort. As you can imagine, the biological samples were collected after the initiation of injection drug use. So it's we're not sure how useful those studies will be with respect to teasing out specifically the impact of ACEs, but many others are working on these kinds of things. You know.
Great. Well, with that, again, thanks so much, Brion, and thanks to everybody who joined us today, and we'll see you soon, and stay well. And as I say, please enjoy the holidays, and we'll all be looking forward to 2021. All right, guys, take care. Bye-bye.